Up next, we have adaptive defenses. And first, we're going to start with a description of the different ways we can explain different types of immunity. So, first, there are four words that we'll use: active, passive, acquired naturally, or artificially. So, I guess 5 words, but whatever. And there's 4 different arrangements that we can make for this. So, we can either say something is naturally acquired active immunity, or we can say something is artificially acquired active immunity, or naturally acquired passive immunity, and finally artificially acquired passive immunity. So what's the difference between all of these words? What does this mean? So the difference between active and passive: active means that you will gain some sort of immunity, but you have to initiate your own immune system in order to get that kind of immunity. Versus passive means that you'll receive immunity from someone or something else's ability to make that immunity. And then acquired naturally or artificially. If you're acquiring something naturally, this means that you are taking this in with your own body with no additional, any additional man-made related things. Versus artificially means that it's going to come from some sort of other source that was probably likely man-made. So now let's go ahead and combine all 4 of those and give some examples. So first, naturally acquired active immunity. So it's active immunity, which means that at some point your immune system has to kick in and actually develop the antibodies to defend yourself against that antigen. And it's naturally acquired, which means that you're able to do it on your own. And so this is exactly what our immune system does all of the time. And as soon as you're exposed to one set of antigens or one pathogen or one disease, you'll develop some sort of immunity towards that disease. So that's what we would call naturally acquired active immunity. The next one, artificially acquired active immunity. So active immunity, again, that you would have to activate your own immune system to teach your immune system how to defend itself against antigens. But it's artificially acquired in the sense that you weren't naturally exposed to the antigen, you were artificially exposed to the antigen. So the classic example here is vaccines, where we can take like the flu vaccine for an example. So in the flu vaccine, there's going to be a really small antigen component of the flu virus itself, small enough that you won't react to it and you won't develop flu-like symptoms, but big enough that your immune system can still recognize it as the flu. And that's going to be buried into a vaccine with a bunch of other components. But as soon as you receive the vaccine, your immune system kicks in and says, oh, okay, I recognize that this is the flu, and I recognize that I need to make antibodies for the next time I might see the flu. Same thing goes for any of the other types of vaccines. It's the same process. And we would consider this artificially acquired active immunity. The last 2 examples, naturally acquired passive immunity. This is where we're not needing to activate our own immune system. Instead of developing our own antibodies, we're able to get the antibodies from somewhere else. And if we're naturally getting it from somewhere else, that means that they're not man-made, they're made from another human. And the classic example here is where when we're receiving antibodies in infancy while drinking breast milk. And the breast milk specifically will have antibodies IgA, and this is one of the primary ways of actually getting IgA into your body. And once you finally get IgA antibodies into your body, then it usually sticks around in the mucosal linings. So that would be an example of naturally acquired passive immunity. You didn't have to activate your immune system in order to develop those antibodies, you just got those antibodies. And you didn't get those antibodies from a lab or from another animal or another species, you got it from another human being. And then the last example, artificially acquired passive immunity. So again, passive immunity, you didn't have to make the antibodies on your own, you just received the antibodies from something or someone else. And artificially acquired means that now we're either making them in a lab or we're extracting it from another species. So the classic example here is if you're hiking up in the mountains and you get bitten by a poisonous frog or snake or spider, and then you get sent to a hospital and you need the anti-venom or whatever the antitoxin that you need in order to not lose a finger or a leg or whatever it is that has been bitten. That antitoxin or that antivenom or whatever it is essentially is going to contain all of the right antibodies to target and defeat the pathogen that you've been infected with. So you completely skipped a bunch of steps where you didn't have to tell your immune system, hey, here's the pathogen, do what you need to do to attack it. We just went straight ahead to, we received the antibodies from something else, and now we can use those antibodies to target the pathogen accordingly. It being artificially acquired, it can either be made at a lab or the antibodies are produced by another species and then extracted from those species and put into a solution that we can accept without having adverse reactions to. Okay, so don't be tripped up at the end of the day. Don't be tripped up by all of these long types of immunity. Be familiar with what does active mean, what does passive mean, what does naturally mean, what does artificially mean, and pair them up accordingly and just kind of brainstorm, like, what would that even look like? What would that even mean for the immune system? And take it from there. I wouldn't get bogged down with just trying to memorize this all like one by one because it's too much. So take it one thing at a time. So up next, we're going to talk about more specifically what it's like when we have this naturally acquired active immunity. How do we actually activate that part of our immune system? And when we activate that part of the immune system, we actually have a more specific term called the adaptive immune response. And it's adaptive in the sense that as soon as you are exposed to whatever the pathogen is, your immune system will work around whatever that pathogen is. So it will recognize the pathogen. It won't force the pathogen to change or to do anything different to the pathogen, but then it will adapt its mechanisms so that those mechanisms can target that pathogen successfully. The adaptive immune response talks about both B and T cells, but here we'll focus on just T cells first. So first we have cytotoxic T cells, which are CD8 cells also. They're called the same thing. These cells, they're cytotoxic, so cyto cell, toxic meaning toxic, so they directly kill cells. Then we have another type of T cell called a helper T cell. They're also called CD4 cells. They do exactly what their name suggests. They help B cells produce antibodies. We also have regulatory T cells, or Tregs for short, and these cells are just floating around and they're constantly just supervising what's going on in the immune system and recruiting other cells if needed. We also have memory T cells, so once the T cell has learned how to help a B cell or how to kill off another cell, it might also turn on other T cells and tell them, hey, you need to remember what this pathogen looks like so that the next time the pathogen comes, you're able to defeat it. So these are all examples of an adaptive immune response. This is ways that we could activate our immune system in order to, and then we would use those T cells and how we would use those T cells. Excuse me. I apologize that these are out of order. This should have come immediately after the this slide right here. But I'm just going to take a second to sit on a soapbox for a second. Vaccinations. This is one of those topics that I have unfortunately little patience for arguing about. There are three things that I can't be really patient in having conversations of. I'm happy to talk about literally anything and have a really thorough conversation and hear all perspectives, but there are three things that I just have little patience for. One is if you think the earth is flat. Two is if you have an opinion about climate change. There's no such thing as having an opinion about what science says is or isn't happening. And then finally, the third one is not vaccinating yourself or not vaccinating your children or even your dog or cat. All of the organizations that are perpetuating this idea that they're not safe, that they cause all these other diseases, that they are made in inhumane ways, et cetera, et cetera. And especially in this time of COVID right now too, it's just unfair to people that are either in completely different fields and have no science background whatsoever, or unfortunately don't have access to any education and can be easily persuaded because people can sound really, really convincing. So I really encourage you all, as you especially enter into the medical field, that you ask really tough questions. Like, there's no harm in asking, like, okay, well, maybe the other perspective, they're onto something. What is this about? But do your own research and be thorough about it first, before really speculating or passing anything on to the next person. That's all that I can ask for. In the meantime, if any of those three topics, like if you want to argue against any of those three topics, I just I just don't have it in me. Literally anything else I'm happy to chat about. Those three I'm really short and straight to the point about those three. If you have any questions about vaccinations, or if you're like, I've heard about this, what about this? I'm happy to have that conversation as well. I actively will try to follow groups on social media and in my news like channels, so that I can get a sense of what all of these groups are thinking and kind of their arguments, just to keep myself up to date and on top of things, just in case something changes. But there's a lot of evidence. There's only evidence that provides or that says that vaccinations do much, much, much more good than harm. So anyway, if you have any questions, I'm happy to chat about it. But if you want to argue with me about it, I'm unfortunately not open to arguing about this topic in particular. Okay, continuing on. So the adaptive immune response, we can kind of generalize it by this flow diagram, which your book gives you. I added some text to it, as you can see here, and we'll go through this one thing at a time. So on the left side, we have the adaptive immunity, and this is where at some point some cell ate a pathogenic cell, and it ate it, gobbled it up, destroyed it, broke it up into pieces, and then took one of those pieces and brought it to its cell surface and said, hey, look at this antigen. What should I do with this? And then we had T cells floating around and B cells floating around. And the T cells were floating around and they noticed this antigen first. So that's why I have usually activated first. And these T cells are floating around and they wait to grab onto that antigen. And once they grab onto the antigen, they recognize, oh, okay, this is either a good antigen and we can keep on going about our day, or this is a bad antigen and we need to set up some sort of thorough response in order to attack that pathogen. And when the T cell recognizes that that antigen is bad news bears, the T cell will go and recruit B cells by taking that antigen in its receptor on the plasma membrane and delivering it to a B cell. And on the B cell, there'll be another receptor, and these two receptors will communicate back and forth, and they'll have this conversation that says, hey, B cell, I found this antigen. Does it look bad to you? And the B cell's like, oh yeah, that does look bad. What should we do with it? And then the T cell's like, I don't know. What if we did this? And then the B cell says, okay, yeah, sure, let's go ahead and do this. And as a result of this communication, then the B cells start making plasma cells, and those plasma cells start making antibodies. And we call this mass production of plasma cells that will each individually produce a bunch of antibodies, clonal selection. So they're cloning themselves to make more B cells that can produce a bunch of antibodies. At the same time, these T cells that went to go talk to the B cells, they can also have other T cells in the area assist in this whole process. So some T cells, like the cytotoxic T cells, can also come in and directly destroy the pathogen. We can also have T regulatory cells floating around and telling other T cells to come to the scene, for instance. Or we can have other cells come and release specific chemical signalers to get other cells to the area, or directly kill off that pathogen. Whenever we're talking about the adaptive immune response, we have to think about a couple of key factors. First is specificity. So if that T cell was floating around and was looking for that antigen, but for whatever reason the antigen was on a receptor that wasn't readable by the T cell, then this entire process would be completely useless and obsolete. Same thing goes for the B cell. If the B cell couldn't recognize what the T cell was presenting to the B cell, then also useless, right? Like then there's no point in having any T helper cells because none of those T helper cells could even actually talk to the B cell. And I know we're talking on really, 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 really small levels, but you kind of have to picture these receptors as having, they're very specific receptors. They will only interact with certain other types of receptors. They will only present antigens in certain ways. And this allows for really high specificity, even though we have only so many cells that we're working with, but they have very specific cell receptors. This also allows a bunch of versatility. So we have, even though we have 4 or 5 different types of cells that we're talking about, they're still able to have multiple different types of receptors, making them even more versatile. At the end of the day, some of these cells can also become memory cells, and so that's super important so that the next time we get exposed to that same antigen, we can attack it the second go-around. Last but not least, we need some sort of tolerance buildup. So this means that the B and T cells themselves, whenever they're learning to become good B or T cells, that they don't learn to attack self cells. Instead, they have to attack non-self cells. And this is really important because if they become tolerant to self-like cells, then that's a good thing. We want them to be accepting of self-like cells. But if they start attacking self-like cells, now you start running into autoimmune diseases where your own immune system is attacking itself. And then that also, again, makes it completely useless and problematic. See you in the next one.